So the estimation of total serum protein. Normally the proteins which are fibrinogen, albumin and most of the globulin, they are produced from the liver. There are certain proteins such as immunoglobulins which are produced by the reticuloendothelial cells. In our body, the normal range of total protein is 6 to 8 grams per 100 ml, and albumin is 3.5 to 5.5 gram per deciliter. Globulin is 1.5 to 3 gram per deciliter, and the normal albumin to globulin ratio is 1.2 is to 1 or 1.5 is to 1, in that range. So the serum total protein, it is estimated using the method Biuret method. So normally all the proteins, it is containing a large number of peptide bond. So when a protein solution, it is treated with a cupric ion in a moderate alkaline medium, we will get a colored chelated complex. It is formed. So this complex is formed between the Cu2 plus ion, carboxyl and amino group of the peptide bond. So this same sort of analogous reaction, it is taking place between Cu2 plus and biuret compound. So this is a biuret compound. Hence the name biuret method. So this reaction takes place between Cu2 plus and any compound which is containing at least two peptide bond or similar group joined directly to carbon or nitrogen atom. And when proteins are reacted, it gives a purplish violet colored product. So this Cu2 plus can also form coordinate bond with a nearby 2 to 6 nearby peptide linkage. 2 to 6 peptide linkage. So when the number of peptide increases, the intensity of color also increases. So a biuret reagent is 9 gram of sodium potassium tartarate. So in the biuret reagent there is a sodium potassium tartarate which is 9 gram. So sodium potassium tartarate is a complexing agent to keep copper in solution. So we are adding with a 3 gram of copper sulfate in 250 ml, zero point 0.2 normal NaOH. So we will mix both of these, and potassium iodide, 5 gram of potassium iodide is added to prevent a auto reduction to 1, and the solution is made 1 liter, zero point 0.2 normal NaOH, and this is stored in polythene bottle. So take 3 test tube, BTS, B for blank, T for test solution, and S for standard solution. Fill 2 ml of water in each of the fill 2 ml of water in blank distilled water, and 2 ml of test serum in T, and standard solution 2 ml standard solution in. To all these add 5 ml biuret reagent. Stand for 10 minutes and take the reading at by using the for coloring photometry. Take the reading at 540 nanometer. So using the blank distilled water, set it as 0. Then using test and standard, measure the optical density. So the concentration of the test solution will be equal to optical density of test by optical density of standard solution into concentration of standard solution. So the interpretation is, if the serum protein is increased, so the serum protein is increased during dehydration. That is when you are not taking adequate quantity of water, or more water is lost. That is like vomiting, through diarrhea, through Addison's disease, or when the level of ADH is low, more water is excreted through urine, and the condition is called diabetes insipidus. Then certain conditions which is called as multiple myeloma, where the myeloma protein is increased, and the when you are looking when you are estimating the total protein, it will reach up to 10 grams per deciliter. So there are conditions where the serum protein are decreased, especially the nephritic syndrome where the kidneys are damaged and large amount of albumin may be lost through urine. Then conditions such as severe burns, excessive bleeding, protein energy malnutrition, when proteins are not absorbed appropriately, that is intestinal malabsorption. So in chronic liver diseases such as liver cirrhosis, the concentration of albumin decreases and the concentration of globulin increases. So the A by G ratio is reversed. So the functions of the plasma proteins are: one is nutrition. So the amino acid from the protein metabolism, it may enter to amino acid pool or to synthesize other nitrogenous compound.